Good morning, Mr. Davigo. Good morning, ladies and gentlemen. Welcome to this academic ceremony, which I hereby open and in which Mr. Michele Davigo, Michele, I think it is, Davigo will defend the academic thesis entitled Heated Tobacco Products, a Reduced Risk Revolution or a New Public Health Threat. Before I invite you, Mr. Davigo, to present the major results of your work and presented in your thesis in about 15 minutes, I would like to briefly introduce the promotion team, the supervisory team, to the audience. The first supervisor is Professor van Schoten, Frederik Jan van Schoten. He's a genetic toxicologist at this university. Then Professor Opperhuizen, second supervisor tobacco control as expertise, also at this university. Then Dr. Remels. He has an expertise. His expertise is in inhalation toxicology, also at this university. And then I welcome Dr. Talhout. She's an expert in health protection and tobacco control, and she's working at the National Institute of Public Health and Environment, better known as RIVM. So welcome to you all. And now I would like to give the floor to the candidate, Dr. Davigo, Mr. Davigo, to present your major results. Please.
Dear Prorector, dear members of the Corona, dear family, friends, colleagues, and collaborators, I will now guide you through the main outcomes of my PhD thesis entitled Heated Tobacco Products, a Reduced Risk Revolution or a New Public Health Threat. This project represented a fruitful collaboration between three Dutch research institutions, so Maastricht University, the National Institute for Public Health and Environment, and the Netherlands Food and Consumer Product Safety Authority. It's quite old news that cigarette smoking is really harmful for humans. It can damage almost any part of our body and is causative of cancers as well as chronic diseases. And the data are very clear, indicating that more than 8 million deaths each year are attributed to cigarette smoking. And smokers can live as many as 10 years less than non-smokers. As the noxious health effects associated with cigarette smoking have been disseminated throughout the society, in the recent years there was a decline in the amount of cigarette smokers throughout the world. And as a response, the tobacco manufacturers introduced on the market new products. And one example is heated tobacco products, or HTPs, which is nowadays a societally hot topic. This is the product design of the most diffused HTP worldwide, which is named IQOS. It is composed of a heating system, which is a pen-like device, inside which a tobacco stick is inserted, and once the device is activated, the tobacco is heated, as the name implies, at temperatures that range between 250 and 350 degrees. And this is substantially lower than the temperature inside a burning cigarette. For this reason, the manufacturers advertise and commercialize these products using claims such as heat non-burn, reduced risk, and smoke-free alternative to cigarettes, implying with these claims that using HTPs is less toxic than using cigarettes. Nevertheless, these claims are currently unsubstantiated, as we do not have enough evidence to support these claims. So almost 5 years ago, we started this journey, and the question was, can heated tobacco products be considered less harmful than cigarette, or are heated tobacco products harmful at all? We tackled this research question from 3 different angles. So we assessed the acute cardiovascular toxicity associated with use of HTPs by carrying out a human exposure study. We assessed the acute pulmonary toxicity associated with exposure to HTP emissions by carrying out in vitro exposure studies on alveolar and bronchial epithelial cells. And finally, we characterized the emissions of IQOS by measuring a wide range of chemicals within their emissions. So now I will give you some more information about each of these three topics. So the human study that we carried out was performed in a close and proactive collaboration with the Mario Negri Institute of Pharmacological Research of Milan. And as you can see from the slide, 3 groups of participants were recruited. We had exclusive cigarette smokers, so people that were only consuming cigarettes. We had exclusive IQOS users, as well as non-users of any nicotine or tobacco product. When the participants were welcomed at our facility, we took the baseline cardiovascular measurement, meaning blood pressure and heart rate, after which, for instance, in the case of cigarette smokers, there was the smoking session in which users were free to use their product. And immediately after use, we measured the post-smoking or T1 cardiovascular measurements. Same design for the IQOS users, with the difference, of course, that they used their HTP. For the non-users, these participants just waited a compatible amount of time for the smoking session of the other 2 groups. So this was a study design, and now I will show you the results regarding heart rate. So these graphs show the heart rate measured in all participants and time points. As you can see, for the 2 groups of users, so the one on the left and the one in the center, the darker line, so either the brown or the purple, is always or almost always higher than the corresponding lighter line. So this is because, by clustering the data together, the heart rate of both groups of users was substantially increased upon product use, whereas there was no increase in the group of non-users. So we then carried out in vitro exposure studies using bronchial and alveolar epithelial cells to investigate the pulmonary toxicity of these HTPs, these new products. To carry out the exposure studies, we produced and also carried out chemical analysis on cigarette smoke extract and IQOS extract. So these extracts were produced by bubbling mainstream emissions from either a cigarette or an IQOS inside a buffer solutions, so to produce extracts that were handy to use in the lab. And in the case of this study, we exposed bronchial epithelial cells for 1 hour, after which we compare the genotoxic potential of cigarette smoke versus IQOS by measuring 2 types of DNA damage, DNA oxidation, as well as DNA strand breaks. And here we go to the results. So as you can see, well, the white bar represents the negative control, so cells exposed to air, to nothing. And as you can see, the orange bar, which represents cells exposed to cigarette, is substantially higher than the blue bar, so there was more DNA oxidation in response to cigarette smoke. On the other hand, though, for the strand breaks, you can see that the higher levels were observed in response to IQOS emissions in vitro. Another exposure study involved primary bronchial epithelial cells. So the difference is that these cells were obtained and isolated from patients from the Maastricht University Medical Center. These cells were exposed for 24 hours, so to resemble a longer exposure time, after which we isolated mRNA and carried out RNA sequencing, which is a technique that allows to really compare the gene expression profile of cells upon exposure to either cigarette or IQOS. And here you can see a volcano plot, which shows the amount of genes that were differentially expressed. So the red dots show upregulated transcripts, so genes that were induced, whereas blue shows downregulated transcripts, so genes that were repressed. And here you can just see that in response to cigarette and IQOS, there was a very similar response in vitro. And we furthermore carried out gene ontology analysis just to assess which molecular pathways were induced or repressed in response to the exposure. And well, these cells had issues to replicate as cell division was repressed. And there was a concomitant induction of pathways that are associated with 2 well-known lung diseases that are chronic obstructive pulmonary disease, or COPD, and lung cancer. One last time to the study design slide so that I can show you how we carried out the chemical analysis on IQOS emissions. So in this case, we included different products. The research cigarette 1R6F, a commercially available cigarette which is Marlboro Red, and we included a total of 9 differently flavored IQOS tobacco sticks. And in this case, the emissions were collected using a smoking machine that allowed us to collect the emissions through a filter pad. And these emissions were then analyzed, and we assessed actually a wide range of tobacco-specific toxicants. And here I will show you the results for nicotine, which is the most addictive compound present in tobacco products, as well as aldehydes and nitrosamines, which are the carcinogenic, harmful chemicals. As you can see here, the levels of nicotine in IQOS emissions were about 50% lower than the smoke of the 2 cigarettes. Also, aldehydes were reduced between 50 and 60% in the emissions from IQOS, whereas only really low amount of nitrosamines were detected in IQOS emissions, actually at levels that were very close to the detection limit. So this was an overview of the results, and now I will try to draw some conclusions from these results. Well, using IQOS may well cause similar acute cardiovascular toxicity as cigarettes, as we showed in our human exposure study. In vitro, we found out that IQOS emissions can damage DNA and can potentially higher the risk of developing lung disorders such as lung cancer and COPD. And finally, although IQOS emissions contain less toxicants, these lower levels may not mean that users are actually exposed to lower health risks than smokers. So what can be the implications and the take-home message of this project? I believe that currently we do not have enough data supporting the claims that HTP manufacturers use to advertise these products. So we believe that these claims just should not be used. And actually, since advertising over cigarettes is not allowed, we don't know why advertising on HTPs should be allowed. And actually, we believe that it should be forbidden. And a word to regulators and political authorities, well, I believe that reaching the people, reaching the users, and let them understand the addictive potential and the health risks associated with the use of these products is really important to safeguard public health. And now I go one more time to the title of my project to just try and answer this question that I raised. So are HTPs a reduced risk revolution or a new public health threat? Well, we do show from our study that IQOS deliver lower levels of tobacco-associated intoxicant than cigarettes, but these emissions are toxic for human cardiovascular and pulmonary system. And finally, what I think is very relevant to underline is that the global market of IQOS as well as other HTPs is growing at an astonishing speed for different reasons. These products are innovative, are trendy, are almost fashionable. They are addictive. These emissions contain nicotine, and the manufacturers can still perform strong marketing campaigns on social media, but also with these IQOS hubs that are disseminated in major European cities where people can try the product and, of course, purchase the product. And this led to the commercialization of IQOS in more than 80 countries, and with a total worldwide amount of users almost reaching 30 million people. Reason why I really think that these products are rather to be considered as a public health threat in the current moment. And I want to thank you for your attention, and I give the word back to the prorector.
Thanks a lot, Mr. Davigo, for this nice presentation and also clear conclusions. Your supervisors have selected a team of experts to assess the quality of your thesis. Your thesis was assessed positive, and that's why you are now allowed to defend your thesis and the work that you have presented in your thesis, because there always remain questions. And that is why the opposition will now be opened by Professor Fransen, He is an expert, he's professor in respiratory medicine at the Maastricht University Medical Center. Professor Fransen.
Thank you very much, Mr. Pro-Rector. Dear candidate, first of all, my congratulations on your thesis. It was really interesting, but also I think it's really important that you independently from tobacco industry studied this topic. So congratulations with that, and also congratulations to your supervisory team. And also the way that your thesis is designed, so to say. It was the first time that I actually caught my teenagers looking into a thesis, probably because there's something that attracted them, and I hope that's not in a bad way. Well, you may know that I'm professionally interested in COPD, which is the main respiratory disease in the world, especially also in the Netherlands, more than 800,000 patients. About 90% of them have a smoking history, but also not every smoker develops COPD. And that's what you highlight in chapter 1 of your thesis, where you say, well, about 20% of smokers develop COPD. That's really important, and that's why I was a little bit surprised if you go to chapter 4 on page 81, where you study the exposure of primary bronchial epithelial cells, the exposure to IQOS and cigarette smoke extract. And I was especially intrigued by the donors you selected for that experiment. Because from the table 1, it seems that you selected ex-smokers without COPD. So why did you choose a population of donors with these characteristics?
Highly esteemed opponent, sincerely thank you for evaluating my thesis, for the compliments to the research as well as to us, to the promotion team and to me. It is definitely a very relevant point what you raised regarding the donors that we selected for our study. We had to work with the samples from the hospital, and there is a very nice collaboration between university and hospital in terms of exchange of samples. And what was available and was collected from these patients was area of the lung that was not affected by tumors, because we're talking about people that underwent surgery. And these cells were isolated from the basal epithelial cells in these people. Of course, the fact that we're ex-smokers is definitely a limitation, or well, it's something that we can discuss on, because of course it would, on one hand, be better to have, let's say, clean lungs or very healthy lungs, not exposed to toxins. But on the other hand, ex-smokers also represent, they have lungs that have been exposed for long term to these toxins. But specifically, we really, of course, focused on isolating and culturing healthy tissue. Mm-hmm. But I agree with you that by going back in time, ideally we would have patients from healthy non-lung cancer patients.
Yeah, I don't agree. Mm. I understand that you selected ex-smokers or smokers, but why did you select donors that did not develop COPD? Because not all smokers develop COPD, and it seems to me that you select a population of donors that are resistant to the toxic effects of smoking, because they were ex-smokers and still they did not develop COPD. Mm-hmm. Yeah. So would the results have been different if you had donors that did have COPD?
So I believe that one of the main take-home message of chapter 4 is the interpersonal differences in response to toxicants. So definitely, it's very likely that the lungs of people that develop COPD had a substantially different gene expression profile, because this is what we assessed in chapter 4. So I actually believe we wanted to carry out this study to assess the gene expression profiles before the disease is actually developed, to understand the molecular mechanisms that lead to the induction or to the onset.
So you suggest that maybe if these donors would have continued smoking, they would have developed COPD?
Well, that is hard to say. What is definitely another important readout of this table is the FEV1 of FVC, which shows whether the lung has already underwent, well, it's already developing COPD or not. And the health threshold is about 70%. And therefore, most donor 2 is very close to that threshold. So I don't think that the lungs of these people were as healthy as the lungs of non-smokers. But I think that having used these samples was relevant as they were healthy tissue, healthy cells.
Okay. One more question relates to chapter 1, introduction, where you mention on page 8 that nothing is currently known about the long-term health effects of the use of these products, especially regarding lung disease. And I was a little bit surprised by that sentence, because already for 3 years, if you look into the international COPD strategy document, the GOLD document, it states that cigarette smoking, but also the use of heated tobacco products, is a risk factor for COPD. So why are you saying that there's nothing available? Or is GOLD just assuming things that are not there?
Definitely not. I think the thing is that these products, specifically heated tobacco products, are on the market for relatively short time, about 20 years. And to really be able to associate a stressor to onset of a disease, you need long-term exposures or, well, epidemiological studies, as well as maybe exposure studies in confined environments to be able to specifically associate. And if on one hand for cigarette smoke is well known that the connection is critical, long-term cigarette smokers have higher chance of developing COPD. For HTPs, it is a probable assumption, but there is not yet the direct line in correlation.
Okay. Thank you very much. I give the word back to the pro-rector.
Thank you, Professor Fransen. The opposition will now be continued by Professor Conklin. He is present online and on the screen. He's a professor of cardiovascular physiology at the University of Louisville. Professor Conklin, the floor is yours.
Yeah, thank you, Mr. Pro-Rector. And Mr. Candidate, I congratulate you on the thesis, well organized, well written, and your presentation was wonderful. So thank you for that. And thank you for having me on the panel. I think one thing you've established is that you seem to support industry's claims that there are fewer toxicants in the emissions of heated tobacco products. And they suggest that this produces milder effects and maybe less harmful. But you say that may not be true. Can you re-emphasize why your research suggests that?
Highly esteemed opponent, sincerely thank you for your compliments, for your relevant question, and also for sharing science and insights at the conference that we attended together. It was a great pleasure. What we show in chapter 7 is, of course, that HTP emissions on a product-to-product comparison emit lower level of certain tobacco-associated toxicants. And what is known from industry-funded but also independent studies is that using HTPs, mostly in the case of switching studies, so smokers that switch to using HTPs, they most of the time have health ameliorations. So of course what is critical about this topic is the need to assess health effects and risk assessment framework without having affiliations with the manufacturers, because eventually what the manufacturers want is users to keep buying the products, and they probably don't, it's not that important whether you use cigarette or HTPs or electronic cigarettes or nicotine pouches, as long as you keep actually making them profits. So of course they somehow hide this with these reduced risk claims. And I believe there is definitely more need, there is need to assess the long-term health implications, because what we did in our almost 5 years framework was to try and characterize the acute toxicity, but indeed the chronic harmfulness is still a question mark.
Okay, that sounds fine. Are there disease outcomes that maybe you should have looked at that would show that HTP exposure produces a different effect?
This is a very relevant point. When investigating acute toxicity, is difficult to translate to disease, although there could be some allergic respiratory reactions that we may have encountered, for instance, in users in our chapter 3. So to investigate disease, we need longer time and also indeed direct associations which are not possible yet. What is also very difficult is to identify populations of users that are exclusive. And what I want to stress is that nowadays the landscape of tobacco product users is never been this variegate. We have people that maybe use HTP in the morning, electronic cigarette in the afternoon, and have cigarettes at night. So in this sense, it's very complicated to directly associate a certain usage pattern with the onset of a disease. We do show in our chapter 4 that by using a disease-specific database that we compared our RNA sequencing data, and we show that in response to cigarette and IQOS, there was induction of genes known to be associated with COPD. But this is, of course, not sufficient because of our exposure study that was limited and simplistic in a way. I hope this answers a bit your question.
Yes, it does. And in fact, some of the acute cardiovascular effects could be attributed to nicotine. Whereas, did you find any other constituents in the heated tobacco product aerosols that you don't find in cigarette smoke?
That's a very relevant question. So in our study, in chapter 7, we only focused on toxicants that are known to be present in tobacco smoke and to be responsible of their addictive potential and adverse health effects. From literature, some untargeted chemical analysis, which are needed to characterize the emissions from a chemical and physical perspective, they showed higher levels of certain, there was one nitrosamine and one polycyclic aromatic hydrocarbon that was at higher level in HTP emissions. What is already known is that HTP emissions contain a much higher level of water and humectants, such as propylene glycol and glycerol. And we believe that these chemicals may well facilitate the transfer of nicotine from the alveoli in the lungs to the blood, potentially resembling addictive threat for users, because indeed these products contain lower level of toxicants, but the release of nicotine is comparable, and we also believe that the absorption can be of concern.
Indeed. In fact, I would also suggest that perhaps topography and use of HTPs could be different from cigarette smoke, and that the more frequent exposure could produce different effects than use of cigarettes. What do you think about that idea?
That's a really good point. And also by carrying out the human exposure study and also talking with people, which is, I think, a very critical way to understand the diffusion of a society, of a product and the reason why people use. A lot of these users were past smokers in the far past, and they immediately said that once they switched to HTPs, or they had the amelioration of pulmonary functionality, they felt better, but that they were using substantially more. The reasons can be variable. One thing is that this product does not cause immediate irritation in the throat. Furthermore, this product is available in flavors combinations, and it's very diffused among youth that use it without thinking about health effects as much as addictive potential. So in this sense, I go back to the last part of my presentation to say, well, regulators, I believe, should better educate somehow the society and young users to let them understand that maybe they're not at risk, as much as risk as when using cigarettes, but that product will be addictive.
Well, thank you, Mr. Candidate. I turn my time back to Mr. Prorector.
Thank you, Professor Conklin. Also, thank you for being present at this opposition, because we realize that the time frame is, at least it's very late, middle of the night at your place. Thanks again. Opposition will be continued by Dr. Kroes. She's an expert in tobacco control at the Trimbos Institute. That's the National Institute for Mental Health and Addiction. The floor is yours.
Thank you, Mr. Prorector, and dear Mr. Candidate. Also, I want to congratulate you and your supervisors with the highly relevant studies that you performed. It's impressive work with impressive conclusions. In light of the shocking results that you showed us already, or discussed it already a bit with Professor Conklin, my first question relates to your statement that there is an urgent need to conduct long-term in vivo studies, because you discussed with Professor Conklin that there might be difference between the long-term health outcomes between HTP products and cigarette smoke. But my question is, would that really make a lot of difference for public health perspective? And do you think that it's ethically and morally justified to expose participants of these studies to, these toxic substances? Or should we better strive for a total ban on HTP products?
Highly esteemed opponent, heartfully thank you for your compliments and your relevant question. So what we did, for instance, in our chapter 3, when we carried out an observational human exposure study, was to specifically recruit users that were already using the product and did not have wish to terminate use. So in this sense, you would not absolutely, I feel definitely against letting people use products that are not already using, for one thing. And regarding health effects, well, what is becoming clear from literature are the acute health effects, which are comparable in case of cigarettes versus HTPs. And indeed, there is still a question mark for the long-term health effects. The question is, of course, it could be that in the long term, HTP users may have lower odds than develop certain cardiovascular or pulmonary diseases than cigarette smokers. But I think what we have to keep in mind is that one thing is comparing cigarette versus HTPs, but what we also have to understand is HTP versus non-use, because teenagers and young adults that today use these products feeling maybe somehow protected by these health claims, by the absence of smoke, think that this is harm-free. But this is not harm-free.
No, but I think that's clear. You showed that clearly. But the question remains, would it make a difference if you can demonstrate that, for example, jumping from the 12th floor is more harmful than jumping from the 10th floor? Because that's more or less the comparison that we're making at the moment. Would that change the message to the user, message to the policymakers? Would that make any difference except for scientific extra knowledge?
I think so, because the users think to jump from first floor, and therefore they think that they are indeed not having they're not exposed to any harm. And if you are efficiently disseminating the results and showing people from a scientific perspective that using these products is comparably harmful in the long run as well, it's not only valuable from a scientific perspective, but also from a societal perspective.
Yeah, well, actually, I do not agree with you. I think that what you showed in your thesis is clear enough. You don't need extra knowledge, I think. I think this is perfectly, there's no doubt about it. You also stated several times that you think that the HTP products are just as harmful as cigarettes. And I think extra knowledge would not add more value to the message to the users. So my question still remains for the final, to wrap up. You think, or do you think it's ethically okay to continue having in vivo studies in human people with these products?
I think that as long as the participants are already using, in my point of view, although of course it would be better to try and let them terminate use, but if the people are already addicted, it's quite complicated to push, and also we're talking about products that are commercially available, so it's hard to forbid, but I'm actually pretty in favor of human study.
Okay.
On people that are already using.
Okay. Thank you very much, and I give the word back to the pro-rector.
Thank you, Dr. Kroes. Also, thanks for joining this meeting, this ceremony. Then the opposition will be continued by Dr. Schins. He's an expert in particle toxicology at Maastricht University. Dr. Schins.
Thank you, Mr. Pro-Rector. Mr. Candidate, dear candidate, first of all, I would like to congratulate you on a very intriguing, interesting book. I've been reading it with very much interest. Also, I have still a young son who was also very much intrigued about the cover. So, but I obviously have a lot of, and of course, I would like to thank also congratulate also the co-supervisors and supervising team for this work. But I have a question, and that follows up on the previous discussion in terms of the in vivo studies in humans. And I found it very intrigued that you have this control group there, but you see a lot of variation there in this control group in terms of cardiovascular readouts, or readout, as I would say. And your study is done in the metropolitan region of Milan. And the first thing I immediately think about that is that there's quite a lot of air pollution there. So I wonder, did you not consider that as a very important factor to include in your study design in terms of at least getting a T0 question there? And also to follow up on that, so maybe you can relate your answer also to what your general opinion is on the relative harmfulness of smoking, whatever you smoke, or smoking air pollution from outside door as a non-smoker.
So, highly esteemed opponent, sincerely thank you for your compliments to evaluate my thesis and also for the very relevant question. So, the study described in chapter 3 was carried out in Milan because our main goal was to recruit exclusive users. So people that were indeed only using the HTP IQOS, actually the specific brand. And it was the most difficult task of the whole study, to be honest, to really find people, then to be also sex and age matched with the other groups. As I said before, the landscape of tobacco users is incredibly variegate. So in Milan is the first metropolitan city where the HTP IQOS was introduced in 2014. And we also had this collaboration with the Mario Negri Institute, and they are expert in epidemiology, so they offered their support in recruiting the specific participants that we wanted. So that's the reason why we carried out in Milan. And eventually it was still hard to to reach the sample size, but we managed. Air pollution does definitely, it's definitely a co-founder. But the fact that all the three groups were, living in the same place, kind of levels up this co-founding feature of the study. The variation that you described is also very important. It's also, I think, a take-home message of the whole thesis, also when connecting with the primary bronchial epithelial cells. What users inhale may be similar, may be the same, but the effects in terms of acute but also chronic toxicity can vary a lot. And we're talking about toxicants that, in the case of cigarette smoke, induce oxidative stress, DNA damage, they are harmful all the macromolecules of our cells. But then if, for instance, a certain person has very effective repair mechanisms, then there will be maybe lower health effects and lower responses upon use. I hope this answers your questions.
To some extent. First of all, I was wondering why you didn't use any paired design in your study approach, because you look at the same person at different time points, and you use all unpaired analyses. And by including the air pollution factor, maybe you could see whether there's some additive or synergistic effect there. Because I think that's also very important to keep in mind. You mentioned earlier that that maybe healthy COPD patients are not exposed to toxicants, but we are all exposed 24 hours a day to that. So, but obviously there's a huge difference there. Now, maybe we can discuss that later in more detail. I want to go to another point, and that's the aspect where you studied the reactive oxygen and the reactivity of your materials. Using a very elegant approach, and you look at carbon-centered and oxygen radicals and so on. And you link that to your genotoxicity aspects. And you have a very nice summary on page 121 about the differences between your conventional smoking, I would say, and your novel product effects. And I wonder whether you could explain that actually the exposure which gives rise to more reactive oxygen species is the one which doesn't give responses in the terms of the DNA base excision repair. Some of these base excision markers are very reactive in this respect.
Yeah, so this was one of the most hard to explain results of the thesis. So what we show indeed that is the cigarette smoke extract contains more reactive oxygen species. These chemicals are the most dangerous because of course they have the ampere electron on the oxygen, therefore they can react with all the macromolecules inside our cells. And they, of course, can cause DNA oxidation. Also, carbon and nitrogen center radicals can cause that, but they do it to a lower extent as compared with oxygen radicals. What you describe in the discrepancy between oxidative ROS and DNA repair, base excision repair expression, may have to do with the timing that we used in our study. The timing was not ideal, meaning that we assess DNA damage after 1 hour, and it is well known that DNA damage can happen almost immediately, actually, up to half an hour, 1 hour, 2 hours. But immediately after the damage happens, the DNA repair mechanism will already rush around to detect the damage and try to repair it, because, of course, what our cells do not want is mutations. So evaluating DNA repair after 4 and 24 hours may have been too late. So this is definitely...
Would have expected to see the opposite then, or in terms of responses? Or?
To see upregulation in the earlier time.
Would you anticipate a more sustained oxidative damage, for instance?
So this is a potential explanation. What it could be, then on the other hand, that we see induction in response to IQOS emissions at the later time points, and it could be that the higher concentration of ROS in cigarette smoke extract cause immediate damage and also fast repair. In the case of IQOS, maybe the damage happened later on, or it continued for a prolonged time, therefore the induction of DNA repair. Maybe you don't agree.
we can discuss about it.
Yes, but I also think that, of course, just to conclude, the assessment of gene expression and protein levels is definitely not sufficient to assess the DNA repair capacity of our in vitro model. And this is, of course, a limitation that I cannot hide.
If I allow to have some more time for another question, if that's still okay. Quick question, quick answer. So may you not have missed something out there because you use a submerged system, you look at part of the whole story. Maybe you can briefly answer on that, what a better approach could have been in this respect, and maybe would have led to complete other results.
Yeah, so the most relevant in vitro exposure system would involve cells grown at the air-liquid interface, ideally multicellular cultures, because we only use monocultures, so also include multiple pulmonary cells, also some immune cells, and expose the cells directly to gas emissions. Also, we only used cells that were exposed continuously to the emissions. The ideal setting would involve repeated sessions of smoking and resting to better resemble the lung of users.
Yeah. Thank you very much. You answered my actual questions very nicely, and I think that would be actually a very nice in vitro approach to follow up on the studies rather than doing additional human studies. And So I would like to give the word back then to Mr. Chair.
Thank you, Dr. Schins. The opposition will now be continued by Dr. Reinhardt. She's expert in respiratory biology at this university.
Yeah.
Reinhardt.
Thank you very much. So I also want to start by congratulate you, Mr. Candidate, with this wonderful thesis. Also, my congratulations to the whole promotion team. It's a highly relevant scientific as well as societal topic that you investigated and that you presented to us also with a lot of passion in your presentation, and I think also in the answers to the questions so far. I actually want to start basically in chapter 2, where you provide a nice overview of the literature of what is currently known about these heated tobacco products. And with respect to pulmonary effects, apparently a lot of research is being done epidemiologically in Korea for some reason, where they find very high odds ratios actually for specifically these products in relation to airway obstruction, airflow obstruction. So you already introduced, like for COPD, that's FEV1 over FEVC. But in these epidemiological studies, of course, this is not necessarily related to COPD. It's especially a population of high school, middle school students. And there's a specific relationship actually also to be found to allergy and asthma. So my question to you is, why did you not study that potential connection any further in your thesis, but decided to focus on the genotoxicity and the potential link to lung cancer?
Highly esteemed opponent, sincerely thank you for your compliments and a very relevant question. So the shortest answer is I couldn't assess everything, and of course we are in the Department of Pharmacology and Toxicology, so we really wanted to assert toxicity to really answer the regulators or provide the regulators with some independent data independent from the tobacco industry. What you mentioned about Korea and Japan is very relevant because these products are very much diffused in these countries. Again, in very young people. And of course, epidemiological studies can be very relevant and should also be carried out in Europe, but they also need quite some time, of course, and you need a pretty robust study population. And here we're talking about thousands or tens of thousands of people. I don't think this could be feasible in the Netherlands because there is not as many exclusive users. And again, I really want to underline the exclusive, because if people use multiple products, then we lose the...
No, if I may interrupt, because, okay, epidemiological studies also just give you an odds ratio, right? You don't know whether you are like looking at a cause or an effect. Maybe because they had certain symptoms, they decide to smoke and they feel better about it. I don't want to offend anybody in toxicology, but it's not, you don't know. So I was actually also a little bit surprised in your overview of the scientific evidence so far that you sort of skipped over any potential evidence in animal studies. Now, I did a little PubMed search, and I found that our colleagues in Rochester, for instance, have tested also the effect of these products in animal models, and they could actually find a relationship to the development of allergic airway disease. That being said, you do have a whole set of RNA-seq data, and I was just wondering whether, aside from the geo-analysis, whether you had a chance to look into individual gene expression or could identify potentially any genes that are relevant to the development of allergic disease and asthma in particular.
So this is also a very relevant point and question that you raised. So I did not specifically go into the allergic reactions because the main idea and also my main interest was to really try and understand the pulmonary effects in the long term. So that's why we also focus a lot on, for instance, describing autophagy and mitophagy, which are well known to be caused by cigarette smoke and be a hallmark for COPD onset and pathophysiology. I do believe, I always describe RNA-seq data as a jungle of data, where you need to be able to find your way through. And of course, we started by having a certain specific research question. By looking back, there is definitely, I do believe that there are some indicators potentially of asthma or other allergic reactions, but also we used cells that are not really directly involved in this.
Oh, I beg to disagree, because alarmin release, for instance, by epithelial cells are major drivers of allergic diseases and the skewing of the immune system. So maybe it would still be worthwhile looking into the gene set.
Yeah, I was just meaning with that we used basal cells.
Still.
Okay, right.
Okay, but since you also switched to the topic of autophagy, mitophagy, ER stress, you know, you look at this in a very acute context, and we all agree we need studies on chronic effects. But is this response that you see in response to the heated tobacco products of induction of ER stress, of DNA response, of mitophagy, DNA repair, sorry, could that not just be a simple physiological response to the stress? Is this necessarily something that predicts toxic effects in the long term?
And that's a very relevant point. So, of course, our in vitro model was simplicistic, and we discussed cells that are exposed continuously for 24 hours to toxicants, to ROS, to aldehydes, to nitrosamines. And, of course, the cells were exposed to sublethal concentration. The cells were not dying, but were, well, at least they were in culture, they were still viable.
Okay,
and of
can
course,
I maybe interrupt and pick up on that just to have a discussion? Because I did also notice with your LDH assay, the baseline levels of cell death were like over 20%. So
in chapter 4,
in multiple chapters, actually, I was happy to see that I think in the BS2B cells, it was much lower, but there was also quite some discrepancy between the LDH and the MTT assay.
Yeah.
So if you could briefly comment on that, and then I'm satisfied.
so about the cell death that was high.
Yeah, it was over 20% with the LDH baseline.
Yeah, so just by looking at 105, page 105, indeed the vehicle was already pretty substantial, and the vehicle were just exposed to PBS, to buffer. I don't know whether the PBS was actually having cytotoxic effect on the cells, but well, in this case, the induction was significant from 4 and 5%, and that's the rationale for including lower concentrations in the bronchial epithelial cells that were, of course, the most precious and valuable samples for the in vitro work.
And with the MTT, it was also not, it didn't seem to be as high of a...
So the MTT was carried out on the on the A549, yes, on the alveolar cells. Of course, MTT is more mitochondrial activity, whereas LDH is a release of this compound in the cell medium. So, of course, the assay can also maybe explain this discrepancy.
Yeah, okay. But I'm satisfied with your answers, and I give the word back to the pro-rector.
Thank you, Dr. Reinhardt. The opposition will be continued by Professor Smeets and finalized by Professor Smeets. He's a professor of clinical genomics with a focus on mitochondrial diseases at the Maastricht University.
Thank you, Mr. Pro-Rector. Dear candidate, my compliments with your thesis, in which I also want to include your promotion team. Very nice work on a highly relevant topic, especially as we thought we were moving to a smoke-free generation. Many scientific topics have been addressed. I would like to start with Proposition 10. Can one of your paronyms read it out for me?
It's only those who do nothing that make no mistakes.
Well, I do not agree. I think doing nothing can be a big mistake. If a child is drowning in a swimming pool, you see it, you can swim, and you do nothing, I consider it a big mistake. And also in the case of HTPSs, would doing nothing not be a very big mistake? And would that mean that your proposition is not true?
Highly esteemed opponent. Well, sincerely, thank you for your compliments and for the very tough question, to be honest. So definitely, I wrote this, which I call actually a preposition in terms from the Latin meaning of it, so place in front. And the reason why I wanted to indicate this is that, well, a PhD is a journey.
It's not about the reason, it's about what you state. Is it true or not true?
That, well, doing nothing is also a mistake in some cases, but then being active, I always more for the proactive than for the staying still.
Okay, so we agree, I think, on that. That brings me back to a more scientifically relevant question. In chapter 6, you use human alveolar epithelial cells 8549. I have some problems with these cell lines. It's a cancer cell line that has multiple chromosomal abnormalities. You put smoke on it, and you want then to say something about mitochondrial function in primary cells. How could that ever be the case?
Yes, so that's definitely a limitation of our study. Ideally, by studying alveolar functionality, it would be ideal to have used also primary cells.
Yep.
The point of these all in vitro chapters was always to have the comparison, cigarette versus HTP. And of course, although having indeed a more cancer phenotype in terms of glucose metabolism, these cells show induction and actually overexpression of respiration that was increased substantially in response to the exposure, and this was consistent between cigarette and HTP. So although with limitations, the functional assay and the functional readout, I think, gives us some relevant potential insights in the acute toxicity.
This would imply that the model would not matter as long as you find differences between smoke and Is that what you're saying?
No, it's not what I'm saying. I'm saying that this model has limitations, but of course we're still talking about an alveolar cell type, and alveolar critical as they are involved in the exchange of gas.
I hope it's still available because it has so many chromosomal abnormalities. I would wonder what type of cell it really is. And it's also not relying on the mitochondrial oxidative phosphorylation. It is relying on glycolysis. So also metabolically, it's different. So would that not also have impact on your conclusions?
So that would definitely change the take-home message, probably. What we did was to really try and understand how the cells were coping with the stressors. Mm-hmm. And again, of course, comparison. And of course, the increased respiration was also a big unexpected result for us because we expected cells to actually respire less. And we believe that this response was an adaptive mechanism to the stressors. Cells were exposed for 24 hours to the emissions, and those that were indeed still alive, and after which we measured respiration, they were probably overactivating their mitochondrial metabolism to actually survive and be viable.
Yeah, that brings me back also, how do you want to validate that indeed? Because you say, I find something, but you have only one cell line, and you already in chapter 4, you show that 3 is already not enough, and I can imagine that in a population-broad problem like this, you would need many, many more. How, you find something, how are you now going to show that it's true, that it's meaningful?
So repeating these experiments on primary cells would be a start. Of course, I'm not a fan of animal study, but it can also be a possibility to use animal models to assess potentially the health effects. But in terms of respiratory capacity, more advanced in vitro models, co-cultures exposed at the liquid interface can represent better the human situation.
And how many would you need? Because you talk in chapter 4, you have a lot of variation between the donors. The overlap is very limited, especially in the gene expression studies. And you have the tendency then to make quick conclusions. You have 2 males and 1 female. Everything which is in the female and not in the males is due to the sex, you conclude. That is a bit, I think, an overestimation. I don't think that all women are the same here. So if you find things in one woman which is different from the man, would you consider then sex-specific? I would not tell. So I think in any way, but that's also in these type of studies, I think the numbers count, and you would like to have simple tests on big numbers which would really support your findings, because the more complex the analysis, the more difficult it is to draw a central conclusion. What would you do?
So this was, of course, a speculation or an hypothesis to explain this gigantic difference in terms of gene expression profiles.
And it's one, so it can be an outlier, it can be a different person.
Yeah, but this was also...
Once the gene expression on Italians and Dutch people, they really separated, and although that was not related to the condition.
It's true. This was, of course, done with cells exposed in the same way, to the same concentrations, and this absence of gene expression profiles in the other 2 donors made us wonder why this is happening. And the only actual difference was the sex. And actually, by looking in literature, there are sex-dependent differences. Our sample size is too small. What is the ideal sample size? Sky's the limit.
Yeah, it doesn't exclude that it's sex different, but you cannot conclude it, that as far as
That's
I
true.
can see. And I was also looking a bit triggered by the way you looked at gene expression on the mitochondrial system, and you say in chapter 6 that complex 1, 4, and 3 are reduced, but you only have one nuclear complex 4 gene you test, and 2 which are mitochondrial DNA encoded, 1 nuclear complex 1 gene, and for complex 3 also 1. Can you draw such a conclusion on a single gene of a complex with tens of genes involved? And you put them also on top, and I didn't really understand why you did that. Yeah,
So you talk about page 168.
yeah, something like that. Yeah.
So the
You can briefly finish your answer.
So of course, the OXPHOS complexes are very multifactorial, and studying certain markers is not sufficient. We worked with the primers that we had available, and also with the time mostly. That was the limiting step. And by assessing, using multiple cell lines, multiple readouts, we tried to cover as much as possible for the in vitro pulmonary toxicity, but there are limitations, I agree.
Thank you.
Thank you, Professor Smeets. Mr. Davigo, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and the quality of your defense. I request that you and your company await the result of our deliberations and our return in this room. Mr. Davigo. The degree committee here present has assessed the quality of your thesis and the quality of your defense. In view of its positive verdict, and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor van Schoten is authorized to confer upon you this academic distinction in accordance with Dutch university customs. I invite your supervisor now to take the floor.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent and responsible?
Yes, I do.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Michele Davigo, the degree of doctor, and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate, signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university. Dr. Davigo, congratulations. You must be very proud what you did today and what you did the last 5 years. At least I'm super proud on you. And as traditionally, the laudatio will be given now, and your daily supervisor, Alex Remels, will do that.
Thank you very much, Frederik-Jan. Dear Michele, Dr. Davigo, it's my honor on behalf of the entire promotion team to be the first to congratulate you on this achievement. I will now try to say a few things in Italian. Caro Michele, Dr. Davigo, congratulazioni per questo grande traguardo. Puoi essere orgoglioso di questo risultato. Desidero fare I miei complimenti a te e anche alla tua famiglia e ai tuoi amici che sono venuti fin dall'Italia per essere qui oggi. Alla tua famiglia, tuo padre, tua madre e tua sorella, vorrei dire che potete essere molto orgoglioso di Michele. I will now continue in English.
Wow.
I still remember, thank you. I still remember the start of the project. Anton, sitting next to me, through the Netherlands Food and Consumer Product Safety Authority, suggested to investigate these new tobacco products that were spreading rapidly. Heated tobacco products, claimed to be safer than regular cigarettes, but a lot was unclear. The plan was to investigate this in a collaborative way together with NVWA, RIVM, and Maastricht University. We posted a position online and received applications from multiple candidates. We made a first selection, and you were invited for the second round and an interview. You made a good impression, and based on that, we selected you for a final round in which you had to make an assignment to present to us. After this final round, it was very clear, together with Anton and Frederik-Jan, your promoters, and Reinske, your co-promoter, we decided very quickly, in fact, immediately after the interview, you were the best candidate, and we wanted to offer you the job. I had the pleasure of calling you, and you already know it's going to come now, to announce the good news. When you picked up the phone, I gave you the good news and said, if you're still interested, the job is yours. It became very silent on the other side of the phone. I waited a few moments, thinking maybe the line was interrupted, and proceeded to ask if everything was okay. After some more silence, you said, sorry, I did not expect this. I had to sit down for a little bit. Luckily, you accepted our offer, and we could organize the start of your PhD. This was the end of 2020. Your position officially started 1st of February 21, a crazy time which all of us still remember. We were in full-blown quarantine mode because of the COVID pandemic. This made the start of your project challenging and very different than we would have ideally liked. It was part of the project that you work at two places, at RVM and at Maastricht. And you started at RVM, which was the most convenient, as at the time you were still living in Wageningen. And RVM had a very strict interpretation of the rules with respect to COVID. And during that time, only people that really needed to be there were allowed there. This meant we had to supervise you online during that starting period. Fortunately, Walter Klerks is in the room today, was there to help you, guide you around the lab, and you started measuring several toxicants and cigarette smoke emissions, and you had to master the use of a smoke machine. It took until late spring that year before we actually met in real life at RVM. We finally got to see you in real life. You were there, very proud, with an Italian flag, because the Azzurri, the Italian football team, had just won the Euro Cup final against England. I'm sure you will. Yeah. Long time ago. I can imagine this was a little bit of a lonely time, an empty institute, working from home, etc. So I was happy to see that after this initial period, you decided to move to Maastricht, the start of a new chapter. You got started on your work here, you quickly learned all the techniques and collected a lot of data. You started writing your first manuscripts. The chemical analysis that you worked on at RVM paper was published very fast. The mitochondrial paper, not so much. But you persevered and we made it. Recently, also 2 other manuscripts got accepted with you as a first author. 2 others are pending as well. And you also have a co-publication with Irene, one of your paronyms, standing next to you today. You can be very proud of this achievement. Another great achievement of your PhD, but not on a professional level, is that you met Christina at our department. I'm happy to see that you are enjoying life together still to this day. You also traveled a lot during your PhD. Of course, a few times back and forth to Prague to visit Christina, but you also went to more exotic locations like New Zealand and to China for the wedding of our old colleague and friend Wembo, where you were the best man. Also, you visited multiple conferences, and I made a very short list, maybe I will forget some. 2022, you went to Baltimore, Society of Research Nicotine Tobacco. After that, we went to San Diego, together 2 weeks in the US, a great time. The year after, Nashville, Tennessee. I'm sure you also remember our time out there. After that, we went to Colorado, the 2 of us, where you attacked the slopes on your skis. 2023, there was a conference in Spain, and last year, Salt Lake City, Utah, also in the USA. Also, as you already mentioned, some time spent in Milano with our colleagues from Mario Negri, where you did the human study. You also supervised multiple students. I saw some of them here today. It's very nice. That helped you contribute to the thesis as well. We would like to thank you, Michele, for all your efforts. You did great, and I'm sure you have a bright future ahead of you. I would also like to thank some other people. First of all, the other members of the promotion team. Thank you very much for all your work and help and assistance. Anton, thank you for the support in this project and others in pursuit of the investigations of tobacco-related products. Without you, this product would not have been possible. Frederik Jan and Reinske, thank you very much for all your contributions. I think I can also speak for Michele that we truly enjoyed our way of working together as a team from different disciplines towards a mutual goal. I think as a team we can be proud of the results we obtained together. Also, I would like to thank all the members of the assessment committee to be here today and for all your time and effort to read the thesis. And a special thanks for Professor Conklin. Dan already mentioned, we only realized later it's the middle of the night for you. So thank you again. To end, Michele, Dr. Davigo, we wish you all the best with the new chapter of your career and your life. You moved to Dordrecht, ready for a new phase. We hope you like it just as much as you did Maastricht. We hope you find a new, nice job that suits all your qualities. We will miss you in Maastricht, but know you're always welcome to visit when you are around. Thank you.
Thank you.
Highly esteemed Dr. Davigo, it is my honor to congratulate you on behalf of the Maastricht University and the Board of Deans with the degree that you have just acquired. I would also, of course, like to congratulate your supervisory team, your family, padre and madre. Congratulations, very proud. And also Christina, congratulations. And of course also all your friends, which I mentioned in the extensive acknowledgements of your thesis. And also those, and several of those are present, I understand, those that have collaborated with you on the different chapters in the thesis and on the publications. And I saw that several of the chapters have already been published, and I'm sure that the others that are submitted will also soon appear in international scientific journals. You have, when looking at the thesis, you have embarked on an ambitious project, but a very relevant study, very timely because of the explosive use of heated tobacco products by mainly the younger generation. Your defense was clear. Your presentation, first of all, was very clear, very nice slides, and it showed that you are a passionate researcher. The term slick for the presentation, which is a positive terminology, was used for your presentation even. The defense, you were confronted with challenging questions, but held up very well. And it was a clear academic defense also. You addressed all the topics, going from basic research to ethics. So you have a broad overview of the field. And I thought you were convincing in your answers, and you remained calm. I'm not sure whether you were nervous, but I got the impression that you started to enjoy it during your defense. The final conclusion is that you're a professional scientist, and I hope that your future will be in science, because you have proven by your work and by your thesis that you're an excellent independent researcher. Before I finalize this, so again, congratulations. Before I finalize this academic ceremony, I would also like to thank the members of the PhD office and the technical staff for organizing this ceremony. And I would like to mention, or to ask actually the audience to leave the room and to go to the, after finishing the ceremony, to leave the room and to go to the place where the reception will be. I'm not sure where that is. But I would like to ask the family to stay here in this hall, because we will make a picture with Professor Conklin in the back, and together with the promotion team and all the opponents. And then we will go to the staircase and make some other pictures. So having said so, I hereby close this academic ceremony.